Hi, everyone. I am Leigh Schmidt, and I am a family nurse practitioner. I work for two companies. I work for Palos Verdes Medical Group as a care provider, seeing patients in the office, just like if you were to go see a doctor or, you're sick or any of that good stuff. And then I also work for our other company, which is called Peninsula Research Associates. Now, let's see. That is the wrong button. Let's see. I cannot for the life of me remember. Here it is, present. My apologies. We're good now. Okay. So I work on two different sides here. I work for both the medical side as well as the private practice and the research side. This is just a little bit of my history, kind of, what I went through to get to where I am now. My background is nursing. I've been a registered nurse since 2002. I like to joke it's since the dinosaurs roamed the earth, and back then we even had paper charting. There were no computers back then in nursing, and everything was handwritten. I got my Master's of Science in Nursing from USC, fight on. And now I'm currently working as a family nurse practitioner and a clinical sub-investigator. And my daughter is Chloe, and she is an 8th grade REN student, also a big Dodger fan, although they've been letting us down a little lately. So let's see here. Let me figure out where to put this screen. So clinical research, what is clinical research? A lot of people are kind of like, what does that mean? You just work in a lab all day, looking at slides in a microscope. And yes, that's definitely part of it for, some different aspects of it. For my clinical research that I do, we work with pharmaceutical companies to discover new treatments, new medications, and currently vaccines are a big hot topic. I know you guys have probably heard a lot about it. Where I work, Peninsula Research, was founded by Dr. Lawrence Scher. That's his picture right there in the middle. Many of you guys actually might be his patients if you guys have ever been here before. He's been an allergist, immunologist, and pediatrician in the area for almost 30 years. And his research company, Peninsula Research, has done over 200 clinical trials. Currently, we have about 15 trials that are open and running right now, and we've got another 10, 15 that are getting ready to start. And there's a lot of different people and a lot of moving parts in clinical research. Doctors like Dr. Scher are the ones who work as what we call the investigator. Nurse practitioners like myself work as investigators and sub-investigators. We have research coordinators, which you can see the girls in the background of this picture here. They work as some of our research coordinators, and you can see the mounds and mounds of paperwork and binders. There is a lot of record keeping in research, and so the research coordinators do a lot of the record keeping, and they also interact with the patients. We also have what we call quality control monitors, and everybody basically works together in collaboration with pharmaceutical companies to try to come up with new solutions to really difficult and complex medical problems. There's a lot of steps in clinical research. We have to follow very, very rigorous guidelines that are set nationally, meaning that they apply to the entire country, so that anytime there is a research trial happening, whether it's happening here in Palos Verdes, it's happening in Boston, Massachusetts, no matter where it's happening, everything follows the same set of rules and guidelines. So you have to make sure that your patients are eligible to participate in the study, and every study is different, and they have different rules and different criteria for what would qualify you to be in the study. Consent is in all caps there, because having a patient sign consent is a really, really important part of the research process. Patients have to be informed of everything that's going to happen to them. What are the potential side effects? What are all the possible risks? What are the benefits? Why would you do this? Why would you not do this? All of those things are talked about and explained in detail before the patient has anything else happen. We always do a physical assessment. We want to make sure the patient is healthy and that we sort of know what their baseline medical conditions are. Study protocols are very, very strict. They are set by the pharmaceutical companies or the company that's doing the research, and we follow them, very, very specifically and diligently. And then administration of an investigational product. That could be anything from giving someone an injection of a medication, having them use a new inhaler, having them try a new pill that they would take, or a new cream or a lotion that you would put on. Those are all considered sort of investigational products. And then we do a lot of follow-up phone calls, and we draw people's blood, and we follow them closely. There's lots of different phases to clinical trials. Phase 1 looks to see, is it safe? Is it safe to give to people? Are we causing them harm? Are we helping them? We want to make sure it's completely safe. Phase 2, after we've determined that it's safe, they move on to phase 2, which is, does it work? Does it do what we want it to do, what we expected it to do? Phase 3 involves basically, can you duplicate those results on a larger scale? If we do this same trial, with several thousand patients, are we going to be able to get the same results? And all of these studies are monitored very, very closely, and you are not allowed, you have to apply to move on to each of the different phases. What are some of the types of trials that go on? Well, any medication that you've ever taken, at some point, there has probably been a clinical trial on it. Currently, the ones that we do in our office, we do clinical trials on food allergies. That's one of my primary roles here on both the research side and the clinical side, is I do a lot of our food allergies. We do clinical trials for asthma. We do clinical trials for eczema, ADHD, EOE, which is a condition that affects your esophagus and part of your stomach. And we also do vaccine trials. And recently, we were doing COVID treatment trials, meaning that we had patients who came in who were positive for COVID, and we gave them a treatment to see if we could help make them better faster. Oral immunotherapy is sort of a spin-off from the food allergy I was talking about. And this kind of talks about sort of the application. So we start off with a clinical trial, and we see whether or not something works, and if it works well. And then if it works well based off of that, it actually will come into practice in your doctor's office. And oral immunotherapy is one of those treatments. So one of our clinical trials that we did was looking at kids who are allergic to peanuts, kids who have peanut anaphylaxis. And we did an entire clinical trial. It lasted for about 1.5 years, 2 years. A lot of it is still ongoing, but based off of that information, we were able to apply that in our clinical setting. And so now on the other side at Palos Verdes Medical Group, we use what we learned there, and we desensitize kids to peanut, meaning we're helping them treat their peanut allergy. And so for me, it's really cool because you see the science in the experimentation phase and where you're trying new things. And then when it works and you have really good data supporting what you're doing, then you can apply it and actually help patients in your clinic. Types of trials we're doing now. Trials are always happening. We always have new trials. So the 2 new big food allergy trials that we're doing, we are going to be starting a multi-allergen study. And what that means essentially is that we are going to try and help kids who are allergic to lots of foods get desensitized to all of them at one time. So if you're allergic to peanuts, and you're allergic to milk, and you're allergic to eggs, and maybe you're allergic to almonds, we can treat you for all of them at the same time. That's what this trial is looking at. So that's really, really exciting because it can save time. Right now, it's like a 6 to 8 month process for each food. So you can imagine if you're a kid who has 6 or 7 food allergies, it's going to take you a long time to get through all of those. So with this, if this works out and we get great data and it's supported, this means you can get rid of all your food allergies at once in one 6 to 8 month period of time, which would be a really big deal for kids that have a lot of allergies. The other really cool study that we're going to be starting, we haven't started yet, is what's called the peanut nanoparticle study. And that one is specifically looking at actually an IV treatment. IV is where you inject something into someone's veins, but that would be a one-time treatment. And so that's really exciting because if we could get rid of someone's peanut allergies in one visit, I mean, that would be truly life-changing for those patients. So those are two of those really exciting studies that we have coming up. COVID-19 vaccine trials, we have several of those going on. We started off in the very beginning with one type of COVID vaccine trial, and now we're doing another COVID vaccine trial, and I'll kind of get into the details a little bit more of both of those, but that's been really fascinating as well. So the first COVID-19 vaccine trial that we started in, we were one of 11 sites across the entire country. It was a phase 1 and phase 2 trial, meaning phase 1, is it safe? Phase 2, does it work? Does it do what we want it to do? And it was run by a company called Sanofi and GSK, and we had over 50 participants coming in to try this new vaccine. And you guys are all familiar with vaccines, right? You guys all have had to be vaccinated prior to starting school. You know, it's the bad part of the pediatrician visit where they tell you, sorry, you got to get shots today. But really important for helping to keep everybody healthy and safe. I hope this video works. I don't know how much you guys have seen or kind of are aware of how our immune system works and why vaccines are helpful. But I wanted to play this very quick little clip just so you can kind of see. It's a really fun little animation that sort of explains at basic level how our immune system works and why it's helpful, okay? So I am gonna attempt to push play here, and please let me know if it's not working, okay? Just give me a thumbs up if you can see that it's playing. Good. Good.
Leanne, Ms. Schmidt, it's a little bit, we lost the sound. One of the things that can help, if you want to pause.
Okay, let's see. Oh, okay, we lost sound.
Just one thing I do is, if from your end, if you turn off your, turn off your camera, put a stop video.
Turn off my camera.
Yeah,
okay.
And then back it up again and start from the top.
Okay.
Seems to work
and
for
just
me.
leave your mic on.
Yeah.
And leave my mic on or off?
On.
Yeah.
On, okay.
Those
Sorry, guys.
I got that knowledge from the kids.
Okay.
All right.
Imagine millions of immune cells, like white blood cells, all on the lookout for specific germs. If they spot something dangerous, like flu, they prepare to fight. When that flu bug enters your body, the white blood cells move in on it. The immune cells arm themselves and then replicate, creating an army of clones. Then they launch powerful germ-killing agents called antibodies, and they tag the germs for disposal. Once the germ is removed, the immune army can disband. But they leave behind memory cells. Their job is to remember the invader and to sound the alarm if it ever appears again. The first time around, it can take at least a week to mount the defense. But next time, the system is prepared. The whole process takes barely a day. It's so fast, we may never have time to even feel sick. A vaccine pre-arms the immune system by sending in a weakened or dead version of the germ, just enough to be recognized. The immune cells mount the defense, and again, you may feel the effect. But because the threat is low, the immune cells quickly disband. However, the all-important memory cells have been created. The immune system's now prepared for the real germ, but without causing full-scale disease.
Okay. So that video kind of shows you a quick and dirty of how our immune system works and how vaccines help us to create those memory cells, right? The memory cells are sort of the backbone of our immune system, and it's the whole point to why we do vaccines to begin with. Now, with COVID, we have seen two different types of vaccines come out on the scene, so to speak. So one of the ones that we worked with was the early on Sanofi GSK, and it was a COVID-19 trial, COVID-19 vaccine trial. And basically what it did is it took the same science that we use every year for the flu vaccine, and it applied it to COVID. So every year when you get a flu vaccine, you get a piece of protein that's on the outside of the flu. And essentially they're all labeled. All of these proteins on the outside of viruses are labeled typically with letters. So for flu, you have an H and an N. And then the number changes. So like, for instance, a while ago, we had a really bad flu season that was what was called an H1N1 flu, meaning those were the markers that we had. I see people smiling. You remember H1N1, it was a bad one, right? And so what they did is they create a vaccine using those protein markers. So for this one, it uses that same technology. Well, on COVID, instead of that spike protein, that little piece that we use, instead of that being labeled an H or an N, it's with an S. And it's just what we call an S spike protein. And so what they did is they combined that spike protein, which is the S protein from the COVID virus, with a chemical that's called an adjuvant. And for lack of a better word, an adjuvant is kind of like when you get the magic mushroom in Super Mario Brothers and it makes you really big and really strong, but you didn't really do anything. That's what the adjuvant does. The adjuvant makes it so that you can take a very small amount of that S protein, but you get a really, really big response from your immune system. And the reason that's important is because you're able to use a very small amount on each person and still get the response you want, meaning you can vaccinate a lot more people with what you have. Because let's just say, easy numbers sake, let's say if normally it takes 100 of those spike proteins to get an immune response, that means we have to create billions and billions of spike proteins in order for each person to get 100. But what if you only needed to give them one? And then you combine that with the adjuvant, which gives it the superpower to give you better response. Well, now you need a lot less and you can vaccinate a whole lot more people, which is really important. So that's sort of what an adjuvant does. It's kind of a fancy word for it reminds me of Super Mario Brothers. It makes your flu vaccine or whatever vaccine you're using bigger and stronger. So you don't have to use quite as much of it. This is just a picture of what I was talking about. So you can see here on the side, this little blue spike is the S spike protein of COVID. This is a very important chemical protein marker that our immune system uses to identify it as foreign and as a problem. And that's what causes us to mount this immune response. And so that S spike protein, it's just that little piece that's used in the vaccine. It's not the whole entire virus. It's not anything that can make you sick. You're not going to become infected with COVID afterwards because it's literally just this small little piece of protein. Now, that was the first one that we started. Now, Sanofi is coming out with another mRNA vaccine. And let me skip ahead. So you guys are familiar with mRNA vaccines, or at least you've heard about them, right? Are you guys really familiar with how they work, or do you want to watch a video on how they work? If you already have gone over this ad nauseum, I don't have to play the video, but if you want to see the video, I can play it. What do you think? I can only see a couple of faces, so just a thumbs up if you want to see the video, or a thumbs down if no, we have had a lot of mRNA vaccine understanding. A thumbs up. Okay, great. And now I'm going to pause my video and I'm going to play.
Researchers have found that the COVID-19 virus begins the process of creating copies by attaching its spikes to a host cell. Those spikes offer a target for vaccines. With the right preparations, our immune system can utilize this vulnerability. A vaccine is used to arm our immune system. It contains messenger RNA, which can direct cells to make specific proteins. The messenger RNA tells our cells to produce COVID spikes. These harmless spikes give our immune system the information needed to quickly detect a coronavirus infection and produce the appropriate antibodies. The new antibodies respond once COVID has entered the body. They attach themselves to the spikes on the COVID virus, rendering the spikes useless, protecting our cells and keeping the virus from reproducing. MRNA vaccines have been studied rigorously and are a safe and effective way to defend ourselves against COVID-19 infections. Research.
Okay, so that is sort of a very quick and dirty of kind of how mRNA vaccines work. And so the big difference between the mRNA vaccine and the other spike protein vaccine is with the spike protein vaccine, we're just putting those little proteins in, right? So you're getting those as a vaccine. That's the spike protein themselves going in. With the mRNA vaccine, it's actually having your cells make the protein itself. So instead of us giving you them from somewhere else, your body just switches over and starts making those spike proteins. And the spike proteins, remember, are how your body recognizes that there's a foreign invader in there and it mounts an immune response. So it's really pretty cool. It has your immune system essentially work for you. Now, we'll just go back a little bit because a lot of people are like, we already have mRNA vaccines, right? We've all heard about Pfizer. We've all heard about Moderna. We've all heard about Johnson & Johnson. Why would we possibly need another mRNA vaccine? And this just comes down to simple numbers. There are, at last count, when I did a Google search, over 7 billion people on this earth who we are hoping to potentially have vaccinated. Now, Moderna has said that they are going to be able to produce 1 billion. Pfizer has slotted to make 1.3 billion. And as you can see, that still leaves several billion people left not having access to vaccines. And so over here on the right, I've listed, these are all the different companies right now that are trying to also come up with their own mRNA vaccine. And when I say come up with their own, that doesn't mean that they're just starting from scratch. Everyone's sharing science with this. That's kind of the beauty of science, is you share your findings. And so we're all using very similar technologies, but because it's new with each company, we're still going through the same rigorous clinical trials. So we currently are getting ready to start phase 1 of Sanofi, which is the company, their mRNA vaccine. And let's see, we saw that wonderful video. Oh no, oh no, we'll get there. Okay, who gets the vaccine? How do we decide who's going to get the vaccine and who's not? So this is where, when you look at research and, the research that we like to use to develop clinical protocols and how we're going to treat patients with medications, the highest level of evidence is what we call a double-blind, placebo-controlled trial. So placebo-controlled just means we don't know who's getting the vaccine and who's getting placebo. Sometimes the placebo is just saline or water, but we don't know. So the placebo is, are you getting the actual vaccine or are you getting just water? Then what makes it double-blind is that neither the patient nor the investigator that's talking to them and is dealing with them knows what they're being given. So if I see a patient and I'm talking to them and we're going through the process, I have no idea what is in the shot that I'm giving them. And so by having it be double blind, that makes it so that you can't have the ability to sort of influence any sort of responses, because for instance, if I really, really feel passionately about having this COVID vaccine come out and I know who's getting it and who's not, I could have the potential to maybe sway people's answers or maybe focus more on what people are saying than what I would do if I have no idea what they're getting. And so basically what it does is it makes it so the data that we do collect is not, is not influenced by anybody else that's involved in the study. After they get the product, or the investigational product as we call it, we see them at regular intervals to check and make sure they're not having any side effects and make sure that they're feeling okay. And depending on what the trial is, we can monitor people for up to a year. Let's see here, what happens next? So with any vaccines and really any clinical trial, these are all the different stages that it has to go to, right? We talked about stage 1, is it safe? Phase 2, does it do what you need it to do? Phase 3 is, can you replicate it with all of these people? And you can see in here at what point things are submitted to the FDA for final approval, and then when it gets approved for patients to use it. Now, I know there's been a lot of talk about the fact that the vaccines that are out right now have been rushed. And essentially what gets rushed is this process here where it says FDA review. You could imagine the FDA is a really, really large government entity. And if anyone's ever had to go to the DMV, I don't know how many of you guys drive and have had to go to the DMV and wait in line. That is essentially what happens at the FDA. Is it's this huge bottleneck, and there's, thousands and thousands of medications that people are trying to submit for approval. So when they talk about the fact that it's fast tracked, what that means is that the FDA is taking them first. They're taking all of their data ahead of the line. They get to jump the line, and they're going to review all that data first because we need it. We need to figure out a way out of all of this COVID mess, right? Let's see. Why is clinical research so important? Well, like I showed you, we help discover new treatments for so many different medical diagnoses. For a lot of patients who maybe have no medical insurance, or they don't have medical insurance that covers things, this allows them access to, brand new cutting edge treatments. It ensures that all the treatments that are given to patients are safe and effective. And as we're finding right now with COVID vaccines, it has the ability to impact the health and safety of the whole entire world. So it really has been a very exciting time to sort of participate in all of this. I think that's about all I have for my presentation. And I think we were going to do some questions, or, oh wait, I have to stop sharing my screen, don't I? Stop share. Okay.
Yeah, back here and go back to me here. Okay, so great. Thank you so much for that fantastic presentation. I learned a lot as well. Hope everybody else here did. So I'm just going to open it up to questions. I did see the Husky shirt in there as well. That was a nice presentation. So good. I was going to comment on that. And you're very skilled with technology compared to me. So, all right. Any questions? Sorry about the camera angle from online or the snowball on here.
I have a question.
Yeah, go ahead.
What do you think about the different claims of the ability of the mRNA to be able to be taken up into your DNA? So pretty much that it can be incorporated into your DNA.
Yes, I've heard lots of people sort of curious and wondering and worried about that. And the truth is, with mRNA, the way that these vaccines work, mRNA in and of itself has a very, very short lifespan. And so essentially, once that particle gets into your cells, it doesn't go beyond the extracellular area. I mean, it's not entering the nucleus. It's not designed to do that. And so essentially it just stays out in the outer area. It changes over the recipe for whatever protein, that cell is designed to use. So when I explain it to patients on the phone, I talk on the phone a lot. I can't show them my really awesome video. And so the way I explain it is essentially, the cells in your body, just imagine, if you will, that you've got cells in your body that just constantly spit out red Legos. That's the recipe that's inside that cell, and it's for it to make red Legos all day long. Well, when you do the mRNA, it gets in there, and the mRNA vaccine essentially reprograms the recipe. So it switches those RNAs from instead of making, red Legos, it now is going to make blue Legos. right? But it doesn't ever enter the nucleus of the cell. They're not designed to do that. And it does it for a very short period of time. And your body for a short period of time will start spitting out blue Legos. And then after that, it stops and the mRNA is gone. And it doesn't, so your body doesn't continue to make those blue Legos, or in this case, it doesn't continue to just always make spike proteins. It only does it for a short period of time. So it's kind of like it just hijacks the technology and then it stops.
Is it right, though, still that the mRNA can be taken up and be reverse transcribed into cDNA?
Not that I am aware of, and you may have more information than I do, but I haven't seen any evidence supporting that yet.
I have a quick question. So you mentioned that when doing these kind of blind trials, it's really important not to disclose whether they're getting, just saline as opposed to the real vaccine. So is it possible that the disclosing that information to the patient or whoever's handling them could compromise the integrity of the entire trial and get someone blacklisted?
I don't know that I would use the term blacklisted, but we definitely, I mean, it's taken very seriously in clinical trials. I can tell you in our office, we take it very seriously. We don't talk about it. There is someone in the office who knows what people are getting, but they have no interaction with the patient, and they don't tell any of the investigators or any of the people who are involved with the patient. And so it is definitely kept blinded for a reason. And the truth is, I think for those of us that are in research, I truly want to know, does it work or not? I truly want to know, is this safe or not? And so for me, I don't want to know. Don't tell me, because I want to make sure that we're doing everything correctly so that once we have that data, we can use it and know that we're doing good work. And then there is a point in the trials, it's called unblinding, and unblinding happens at a certain point in every single trial. And depending on what it is, it's either sooner or later. I want to say with the vaccine trials, because we knew early on with our flu-like one, the first one I talked about, that's just the straight spike protein, that was pretty early on. And we knew that mRNA vaccines were coming out, and we didn't want, and the people who were running the trial, we didn't want patients to not know what was going on. Because if, for instance, they were given placebo, we wanted to make sure that they had the opportunity to get the mRNA vaccine once it was available. So I believe that one was unblinded at like 3 or 4 months. We unblinded it pretty early on because we wanted patients to have the opportunity to get another vaccine if they were given placebo. So I feel like it was pretty, the interest of the patients and science are sort of always on the forefront of our mind.
All right, I think we have time for one more question. Anybody from in here have a question?
I sent a question in the chat.
Okay.
Let's see. The vaccine for adults and 15 and younger. So far they've just put out just recently within the last, several days that it's now given to ages 12 and younger. I do not believe that there is a difference between the vaccine or the dose. I believe it's exactly the same. And there's no, whether you get Pfizer or you get Moderna, you do want to stick with whichever one you get. So if you got a Moderna, your first Moderna needs to be also a second Moderna. Same thing with Pfizer. But even with those, the amount that you're given is the same. It's essentially just giving you what we call a booster. So similar to, like, you guys have all had tetanus shots before, and every 10 years we give you a booster shot. And that's just to remind your immune system, hey, be on the lookout for these guys. And so with this one, what they discovered is that essentially they needed 2 doses in order to get the full antibody, full immune response that they wanted. In terms of the information on children's vaccines, they are getting ready to start. Lots of places have already started pediatric trials. I know that there's pediatric trials happening in Arizona. Here at our office, we will be starting a pediatric trial for the younger ages. I believe it's 6 months to age 2. It hasn't started yet, but we are in that process of looking at applying these vaccines and the vaccine technology to children as well. And again, all of this is based off of current data. As you guys saw, all of this is it's evolving and happening in real time, which is really hard for science. I mean, think about it. So you guys just did a STEAM fair, right? You guys had experiments that you conducted. How many of your experiments worked exactly perfectly the first time you did it? I'm hoping not very many, because science is mistakes, right? Science is you try it and it doesn't work and you go back to the drawing board and you think about what went wrong and you do it again. If you guys have ever watched any of those videos on that crazy guy who worked for NASA, who does everything out of elephant slime, I mean, most of his failed, right? He failed, and then had great success. And the tricky thing with all of this with COVID is that this has all been happening in front of the entire world, right? Science has been trying to figure this out with all of the trial and error that goes into this, but it's happening in real time in front of everyone, which is really scary and really hard. It's hard for science because they're trying really hard to figure this out, and it's hard for the general population because we're like, why can't you figure this out? Why don't you know? And I think, unfortunately, this is just inherent to science, right? There's trial and error in all of it, which is why we do clinical trials, is because we want to make sure before we start giving things to people that it is safe and it's going to be effective.
Thank you so
I
much.
think that answered.
That's a great presentation. Thank you so much for all that you do and for the presentation. So informative, so educational, entertaining. So I have to send the students out into the wild now, but they really great job. So we'll bid you adieu. Okay, take care.
All right, thanks,
All right.
everybody.
All right.
Thank you.
Thank you. Bye.
Hope we can hear them. All right.